Hello, I'm Alireza. As part of my PhD, I developed a novel experimental technique that uses nuclear physics in order to study lithium diffusion in solid materials. Using this technique, we were able to resolve several questions regarding lithium diffusion in materials such as rutile titanium dioxide, which is a crystal, and these materials could be used in the next generation lithium-ion batteries.
I studied the regulation of swarming motility in the pathogen Pseudomonas aeruginosa, which causes lung and hospital-acquired infections. My findings showed that swarming, a multicellular movement that enables pathogens to spread over surfaces, is a complex behavior that increases antibiotic resistance and influences disease virulence.
Dr. Chu improved the process of using computers to extract meaningful information from biological sequences, such as DNA. He designed computer programs to store data in probabilistic data structures, which purposely store data as approximate signatures in order to surpass the memory and speed limits of representing data perfectly.
I assessed the social and economic outcomes of an individual transferable quota ITQ fishery in British Columbia. My research identified significant failings of the ITQ management system. This research raises important questions about the design and implementation of ITQs and their negative impacts on fisheries and communities.
Hello. During my PhD, I used state-of-the-art nuclear models and statistics to study the imprints of the nucleus on light from exotic atoms, in which the nucleus is orbited by a muon instead of an electron. My work sheds light on recent experimental discrepancies and helps to illuminate our understanding of the interplay between the nucleus and light.
Hi, my name is Mariam Khalfala. I studied the development of marine fisheries in the countries of the southern Mediterranean as well as in the countries of the Arabian Peninsula. My research shows that more accessible scientific tools can be used to better assess the state of marine ecosystems and better manage fisheries resources in data-limited and economically developing regions.
I developed a sensitive method to study how the composition of the chromatin in normal blood cell precursors changes during the blood cell production and how it is altered in certain types of leukemia. My research advanced our understanding of molecular processes that regulate the production of normal and malignant blood cells.
I did my research at the intersection of physics and medicine and developed new techniques to probe the tumor microenvironment. The most promising of these techniques is called oxygen-enhanced MRI, as it supports the delivery of chemotherapies targeted at tumors whose oxygen status makes them particularly difficult to treat.
I studied responses to salmonella infection in macrophage activation states, including the tolerant state seen in sepsis. I identified differences in gene expression that may account for different levels of resistance to salmonella. This could help us to improve the ability of macrophages to resist infection and treat sepsis.
I developed two radiation therapy modalities, known as trajectory-based treatment, where the couch moves continuously. This work introduced a novel algorithm for treatment planning, which can accurately model proposed treatment modalities. This method can achieve plans superior to those generated by standard treatment planning systems.
In my thesis, I described the role of CD45 in regulating red blood cell progenitors in spleen, which protect the mice from inflammatory bowel disease by interacting with other cells. This work shows importance of CD45 as a modulator of immune responses and suggests a new therapeutic approach to treating systemic inflammation that is associated with IBD.
I examined two MRI techniques in the context of myelin health and multiple sclerosis. Through simulations, studies of tissue samples, MS, and control populations, I linked the MR measurements to the biology of MS. By establishing the robustness for probing myelin health at different field strength, I addressed the key issue for using MRI for MS.